All right, cool. So, this here. All right, cool. So, hi, my name is Gary, and I'm a grad student in Bing Ren's lab, and today I'd like to talk about how our lab is using chromatin signatures to annotate functional regions of the human genome. So the DNA in our cells is packaged by histone proteins, and histones are condensed into chromatin to form our chromosomes. So what's interesting is that histones have tails, and that these tails can be post-translationally modified. So for example, we now know that H3K4 trimethylation generally marks active promoters, whereas H3K27 trimethylation marks inactive promoters, and the presence of both of these marks at the same time generally marks poised promoters. There's over 100 different chromatin modifications known to date, and each one offers a different dimensional view of the human genome. A single genomic locus can contain many different chromatin modifications, and the number of possible combinations is huge. And we think that the epigenetic code is an abstraction over the genetic code, and that using it to find functional regions of the genome may be easier than finding them using the genetic code alone. So along these lines, our lab has recently shown that enhancers are typically marked by a monomethylation signature that lacks trimethylation, and that this is opposite to promoters, which contain strong trimethylation but weak monomethylation. And what's interesting about this is that not only are these patterns descriptive of enhancers and promoters, but they're also predictive. So regions of the genome that look like this first signature are generally enhancers, and regions of the genome that look like the second signature generally are promoters. So now that we know that at least two different kinds of chromatin signatures exist, one at promoters and one at enhancers, the next question that we asked is, what are the other commonly occurring chromatin signatures that exist in the human genome? And along the lines of that, what are the functional consequences of these chromatin signatures? So to do this the right way, we require an unbiased de novo pattern finding method that's given only the chromatin modification data alone without any preconceptions of what kinds of chromatin signatures you'd expect to find. This problem is extremely similar to the standard motif finding problem that we all know is defined over sequence space. So the approach that we take is to extend motif finding to histone space. The approach that we came up with is called ChromaSig, and here's how it works. So we're given a lot of chromatin modification data. In this example, I'm only showing two. And the first thing we do is select some interesting regions. So these regions may contain chromatin signatures, and they may be aligned, but they may not even be in the same orientation. So for example, interesting regions A and B here by eye look like they have the same chromatin signature, but region C by eye doesn't. But if you take region C and you flip it left to right, and then you shift it slightly, you'll find that region C does have the same chromatin signature as A and B. So to find these chromatin signatures, the first thing that we do is we define a seed motif consisting of very well-aligned regions. And in this case, it's A and B. So after we find the seed motif, we attempt to align all these interesting regions to the seed. And so in this case, we sampled C, and we list out every single possible sub-window in these regions in both forward and negative orientations. And then we try to align it to the motif. And if we get a good alignment, then we include region C into this motif to refine the motif. And if we don't, then we exclude this region. We then repeat for the remaining regions to align to the motif, and then we try new seed motifs. So at the end of the day, the output that we get is a collection of motifs. Each motif consists of a set of aligned and oriented regions. And one important detail that I left out in the beginning is how do we select these interesting regions? So if we're only interested in promoters, then our interesting regions can just be the promoters, or they can be any other set of your favorite loci. Or if we want an unbiased look of the human genome, and we want to find new signatures marking unannotated sites, we can just let these interesting regions be places with extremely high enrichment of histone modifications. So we applied this data, this method to genome-wide maps of 20 histone methylations. These were mapped by Keisha Zhao's group last year, and they were mapped in CD4 positive T cells using ChIP-seq technology. So we first applied ChromaSig to the 20,000 human promoters, and we recovered 14 different clusters. In this plot here, there are 20,000 lines, one for each promoter, and the 20 different dimensions of histone modifications at those promoters. And so you can see that ChromaSig gives you a global view of the histone modifications that occur at all the promoters. So each one of these 14 clusters has a distinct chromatin signature. Some of them are very distinct, like P2 and P4, which are completely different. And some of them are very subtly different. So, for example, P9 and P12 are actually enriched in the exact same chromatin modifications, but they have a different pattern of enrichment. So we also noticed that there's a very high degree of redundancy at these promoters. And so, for example, these 5 chromatin modifications are either always present together or always absent together. And we recover the known patterns that we should have recovered. Repressed promoters marked by H3K27 trimethylation, active ones with H3K4 trimethylation, and poised ones containing both of these marks. Correlating this to gene expression, we find that chromatin modifications essentially split these places into three different classes of expression. So there's lowly expressed genes, moderately expressed genes, and highly expressed genes. The repressed and poised promoters generally fall into the lowly expressed class, whereas the actively marked promoters are generally moderately and highly expressed. So we can see that small differences in chromatin structure can have big changes in the way these genes get expressed. Next, we moved on to enhancers. So using our chromatin-based enhancer prediction strategy, we predicted about 35,000 enhancers, and the vast majority of these are very far away from promoters, which is what you'd expect. To validate these enhancers, we looked at 2 different hallmarks of enhancer activity, which is DNase I hypersensitivity and sequence conservation. So we find that almost 50% of these predictions are recovered by DNase I hypersensitivity, and at random, we'd only expect about 5%. And 75% of these predictions are recovered by some combination of DNase I hypersensitivity and sequence conservation. Applying ChromaSig to these places, we find 11 different signatures. As expected from our method of finding enhancers, we find very strong enrichment of H3K4 monomethylation at these places and much weaker H3K4 trimethylation enrichment. Like promoters, we see that enhancers are extremely highly redundant in terms of the histone code. And finally, we see that monomethylation of H2BK5 and H4K20 almost always occur together, and these almost never occur with H2AZ, indicating that these 3 chromatin marks may partition enhancers into 2 different categories. Okay, so we've applied ChromaSig to promoters and enhancers, and we found different chromatin signatures. So we could also easily apply it to any other set of loci. But let's skip to why we designed ChromaSig in the first place, which is to find novel signatures marking unannotated sites. So to do this, we located regions with strong chip enrichment of histone modifications that were very far away from all of these other known sites. And in total, we found about 50,000 sites. So applying ChromaSig to these sites, we find 7 different distinct chromatin signatures. And the most striking feature is strong H3K36 trimethylation in U1, U2, and U4. And we also see some clusters that have repressive marks enriched. Now, going back to these first three groups, H3K36 trimethylation is known to mark the 3' ends of genes. But all of these sites are very far away from 3' ends of genes. So they must be marking something else. We noticed that 90% of U1 and U2 are actually inside gene bodies, and that 50% were located near exons. So to see if there's a chromatin signature at exons, we then looked at these overlapping exons. And we can see that for U1, U2, and U4, there's a very strong enrichment of H3K36 trimethylation right at the 5' end of the exon, and that this decreases as you go into the body of the exon. And U3 is the negative control that doesn't have any enrichment. And so looking at this, you can say that there is indeed a chromatin signature marking the 5' ends of exons. But if we take a step back and think for a moment, that sounds like a very strange statement. How can a chromatin signature mark exons when exons are only involved later on in splicing? So when we think of transcription and splicing, we typically think of the model where these 2 events are decoupled. And so if you can imagine this piece of DNA, this gene, there's 2 alternatively spliced exons that are colored. RNA polymerase transcribes this to give you a pre-mRNA that has both of these alternatively spliced exons. And then this pre-mRNA is shuttled to some other part of the nucleus, and the spliceosome preferentially assembles at some of these sites, and then you get splicing to give you a mature mRNA with only one of the alternatively spliced exons. So if there's a chromatin mark at an exon up here, it's very hard to think how it can get propagated all the way down. But in a more recent co-transcriptional splicing model, these 2 events are very tightly coupled. And so spliceosomal factors found on the polymerase actually direct the assembly of the spliceosome on the exon as the pre-mRNA is being transcribed. But still, given this model, it's unclear how some exons preferentially get spliceosome assembly and others don't. But if a chromatin signature is found at the exons, maybe it's the reading of this signature that helps to direct the preferential assembly of exons, of these spliceosome factors on the exons. And so if this model is true, then it offers one way to solve the problem of the extremely highly redundant nature of the sequence of DNA sequences that actually govern splicing. So if this model is true, we'd expect that exons that are marked by the chromatin signature to be much more likely to be included in a mature mRNA than exons that aren't. So to test this, we examined exonic expression of marked exons in pink versus unmarked exons in green. The overlap is shown in brown. And we see that for U1 and U2, these marked exons are much more likely to be included in a mature mRNA than the unmarked exons. And this observation is significant. U3 is the negative control that shows no preference, which gives you a low p-value. And then, but when you look at U4, you see something interesting too. You see that these marked exons are actually preferentially excluded from mature mRNAs. And so we can get a clue for why this is happening or how this is happening at the chromosome signature level. So U1, U2, and U4 all contain H3K36 trimethylation. But U4 is the only one that contains repressive or heterochromatic marks that are bound to it as well, like H3K9 trimethylation. And so maybe H3K36 trimethylation marks active exons, and then addition of repressive marks turns that active exon into an inactive exon. So in summary today, I've shown you, well, these are the chromatin signatures that I've shown you recovered by ChromaSig, and in total they span about 100,000 different genomic loci. With the 20 different chromatin marks that I'm studying here, you'd expect about 2 to the 20th or 1 million different possible combinations of chromatin signatures. And the fact that we only find about 30 here indicates that the histone code is extremely redundant. We recover well-known patterns at promoters, as well as less studied patterns at enhancers. We also recover an unexpected pattern at exons that likely, that indicate that co-transcriptional splicing may occur at a large scale and that it may be directed by chromatin. And also, finally, we also recover novel patterns marking sites of unknown function. That these sites share the same common chromatin signature indicates that they may share the same function. But the challenge now is to find what function this is. So to close, I'd like to thank my advisor, Bing, and my co-advisor, Wei, and as well as the rest of the Ren Lab for all their support and critiques. I'd like to thank my funding sources and the organizers for letting me speak today. Thank you. Questions?
So did you look at, it wasn't totally clear on the analysis of inclusion in mature exons, so of mature RNA. So you're talking about alternatively spliced exons that are included in that particular, in the tissue that you're looking at?
Yeah, there's only one tissue that's examined here, so that's CD4 positive T cells. Yeah.
And do you control for position within the gene or?
No, so like the way we looked at it was if an exon is marked in one gene, we only look at the alternatively spliced exons within that same gene to make sure you don't count for other things outside of it.
Right, but was there any bias for with the marked exons? With the marked exons, you sort of randomly distributed across the gene, or were they found preferentially in certain parts of the
I
gene?
didn't look at that, yeah.
Anybody else?